Welcome. I'm Diana Friedman, Director of External Affairs at Research to Prevent Blindness. Welcome to RPB's Lunch and Learn Eye on Geographic Atrophy and Dry AMD. Today's event is generously sponsored by Apellis Pharmaceuticals. Research to Prevent Blindness is the leading nonprofit organization supporting eye research directed at the prevention, treatment, or eradication of all diseases that damage and destroy sight. We were founded in 1960, and over our 62-year history, we've awarded more than $397 million in research grants to the most talented vision scientists at the nation's leading medical schools. As a result, RPB has been associated with nearly every major breakthrough in the understanding and treatment of vision loss. In today's educational session, you'll hear from two RPB-supported talented scientists about geographic atrophy and dry AMD and its causes, impact, and exciting cutting-edge research. One of our speakers, Dr. Jayakrishna Ambati, has received multiple RPB awards. This includes the Lou R. Wasserman Merit Award, the Physician Scientist Award, the Senior Scientific Investigator Award, and most recently, the RPB David F. Weeks Award for Outstanding Vision Science Research. And our other speaker, Dr. Catherine Bowes-Rickman, is also a multiple RPB awardee. She's received the RPB Career Development Award and the RPB Catalyst Award for innovative research approaches for age-related macular degeneration. We'd like to thank all of the generous supporters of RPB. You make our work possible. The importance of supporting medical research has never been so apparent. We'd like to thank you for your partnership in supporting innovative vision research to ensure that new breakthroughs are possible. And if you've not yet supported RPB, please consider doing so to help make sure we are able to continue these breakthroughs. So getting back to today's session, let me introduce our moderator, Dr. Federico Grossi, MD, PhD, who is the Chief Medical Officer at Apellis Pharmaceuticals. Apellis develops transformative therapies for people living with a broad range of debilitating diseases. The treatments are designed to control complement, part of the body's immune system. Dr. Grossi has worked for Apellis since 2010. Prior to that, he served as the Clinical Research Director for Potentia Pharmaceuticals. He received his medical degree from the University of Cordoba in Argentina and his PhD in physiology from the University of Louisville in Kentucky. Following his postgraduate training in surgery, Dr. Grossi joined the Plastic Surgery Research Laboratory at the University of Louisville. He developed extensive expertise in microsurgery and composite tissue transplantation. While there, he was a crucial member of the Louisville face transplant team, whose publications are now considered hallmark publications in this field. Today, Dr. Grossi will introduce our two speakers, and he will moderate the Q&A session at the end. Each speaker will speak for 20 minutes, and I will serve as timekeeper. Speakers, I'll give each of you a warning at the 18-minute mark. We want to make sure that we have time at the end for a robust Q&A. So Dr. Grossi, please take it away.
Thank you, Diana. I also want to thank the presenters, Research to Prevent Blindness, for organizing the event, and to all attendees. So today we're going to be discussing geographic atrophy and dry age-related macular degeneration, which is a very important topic. It is important not only because of the number of people that it affects, but also the impact it has on vision and quality of life, mostly in the elderly population. So AMD, dry and wet, is the leading cause of permanent vision loss in people over 65, and the risk of developing late AMD actually goes exponentially up with age. So there's about over 1 million people with GA in the United States, with 5 million globally. And while we have therapies for neovascular AMD or wet AMD, there are currently no treatments approved for GA. So the cause of GA is multifactorial, and we'll see some of that today. With numerous environmental and genetic factors, but the dysregulation of complement is believed to play a critical role in the development and progression of dry AMD. And that's why it makes this a very important topic for Apellis, as Diana mentioned. We hypothesized back, in 2005, 17 years ago, even before the publications on, on the genetics, the seminal papers on genetics came out, that complement was involved in macular degeneration. And then we were the first group to test complement inhibition in AMD a few years later. So after all these many years, we have been able to demonstrate that there is a biological effect of complement inhibition in GA. And this is not only important, for Apellis and us as professionals, but also for the scientific community and the patients. All this adds to the body of evidence on the biology of dry AMD and put us one step closer to, potential treatment for GA. So before I pass it to the first speaker, some housekeeping items. I'll remind everyone that we'll be taking questions at the end after both speakers finish presenting. You have a Q&A option in the toolbar, so go look for it. And if you have any questions during the presentation, just add them to the Q&A section, and then we'll keep track of that. And you can also add questions at the very end. So now let me introduce the first speaker, Dr. Jayakrishna Ambati. Dr. Ambati, it's an MD, is the DuPont Gary Third Professor and Vice Chair for Research and founding director of the Center for Advanced Vision Science at the University of Virginia. He's a vitreal retinal surgeon whose laboratory studies neuroinflammatory diseases. His lab has reported numerous seminal advances in journals such as Nature, Science, Cell, and the New England Journal of Medicine. He was selected a fellow of the National Academy of Inventors and is the only ophthalmologist to win the NIH Director's Pioneer Award. He has numerous prizes, including the David Weeks Outstanding AMD Research Award. Dr. Ambati also served on the editorial boards of IOVS, Ophthalmic Research and Translation Vision Science and Technology, and is listed in Best Doctors in America. Dr. Ambati's presentation today is A Journey to End Macular Degeneration. Jay, please go ahead.
Fede, it's great to see you again and to be in this lovely forum with my friend and colleague, Kathy Bowes-Rickman. And thanks, of course, most importantly, to Research to Prevent Blindness for organizing this continuing series that I think is tremendously valuable to the public at large. So let me first start by disclosing that I'll be talking about some technologies in this presentation in which my employers, my current university and my former university have intellectual property interests. I've co-founded several companies. The one that's most relevant to this talk is Inflammasome Therapeutics, which is commercializing some of the technology I'll be describing here. And I think it would be remiss to not point out that 20 years ago, we had a wonderful collaborative research engagement with Potentia Pharmaceuticals, now Apellis. And I really want to congratulate that entire team, which we worked with in Kentucky, for bringing their product so far, so fast, and to the cusp of FDA approval. So congratulations and a big shout out to all my friends at Potentia, now Apellis. All of us, like Dr. Rickman and the hundreds of other scientists around the world who've been working to end macular degeneration, are eager to see therapies developed for this condition. And over the past couple of decades, we've had the great fortune in our generation to see what used to be a clarion call for blindness, which was wet AMD, actually now have multiple effective therapies. And we're hoping that companies like Apellis and several others will come forward to make similar advances in dry macular degeneration. So I'm going to be describing a little bit of this journey and some emphasis on our journey to end macular degeneration over the past couple of decades. And it's with great joy and pride that I thank Research to Prevent Blindness for accompanying me personally and my lab on this journey over the last 2 decades with their tremendous and truly invaluable support. So thank you to RPB for that. First and foremost, we see the world, we interact with the beauty of the world through our eyes. And when we lose this most precious sense of vision, as can happen in the late forms of macular degeneration, it can fundamentally alter the very way in which people live their daily lives. And societally, it's an epidemic, a silent one, but it's nevertheless an epidemic with some 200 million people around the world affected with macular degeneration. It far outstrips the number of people who have Alzheimer's and most solid cancers put together. And with the aging of the population around the world, these numbers are going to only increase even more. But it's not just a disease of the elderly. For example, in our country, a 50-year-old woman has a far greater risk of developing macular degeneration over a 5-year period than she does of developing, say, breast cancer. So what is this macula? Quite simply, it's prime real estate at the back of the retina. It's this super tiny area that affords us the opportunity to experience the beauty of the world in high definition. And when this normal macular appearance goes awry in a disease like macular degeneration, which comes primarily in 2 flavors, the so-called wet form and the so-called dry form, there can be anatomical and therefore functional disruptions to the architecture of that precious back of the retina. And the wet form of macular degeneration, which is characterized by a pathology called CNV, or choroidal neovascularization, is typified by the image that you see at the top there, the ectopic invasion of abnormal blood vessels that lead to bleeding and fluid leakage that obviously can cause vision loss. And the dry form, which is far more common than the wet form, ultimately leads to these large areas of loss of cells in the retina, cells in the retinal pigmented epithelium, the photoreceptors, and the underlying choroidal vasculature, the choroid capillaris, all undergo degeneration at various rates. If there's one good thing about AMD is that it progresses slowly in most people. This is a disease that develops probably over several decades, as these images that Ron Klein was kind enough to share me show. The top shows a 15-year journey of a person's eye from normalcy to early dry AMD to wet AMD. And another patient over a similar time frame going from normalcy to dry, intermediate dry, and then finally geographic atrophy. And fortunately we now have therapies that can intervene at this last culmination stage of wet AMD that can still improve vision in a sizable number of patients. And we're hoping to be able to do that, we as a collective group, in the context of dry AMD. And as I mentioned now, some 18 years ago, we had the first introduction of anti-VEGF therapy that was approved for use in wet macular degeneration. And it was rightly termed a miraculous therapy. These drugs that you see at the bottom all target vascular endothelial growth factor or some related molecules in the case of Vabysmo. This anti-VEGF drugs on a statistical basis improve vision on average for the first 1 to 2 years at least. And it's been a remarkable sea change in how we manage this condition compared to the many decades before. And what you can see is that actually there was an astonishingly compact timescale moving from lab to label. From the time that my good friend, the late David Hinton and collaborator, described the presence of VEGF in human choroidal neovascularization specimens, which was in 1996, to the time that we not only had a good understanding of what VEGF was doing, laboratory studies, toxicity studies, pivotal trials, within 8 years, which is remarkable, we had the first anti-VEGF treatment that was approved for this condition. And subsequent years saw even better therapies, such as Lucentis and Eylea being approved, and more recently, other drugs as well. And so one of the highlights of this that I think bears mentioning is that all of this was done because of a remarkable effort by people around the world in trying to really understand what is VEGF, What does it do? How is it involved? Let's figure it out and then develop rational therapies based on this understanding. And we as a community and we as a population have been rewarded richly for these efforts over the past couple of decades. In contrast, here are now all the approved treatments for geographic atrophy. So why don't we have a treatment for GA yet? Is the problem too hard? I don't think so. Are we attacking the wrong problem? Perhaps. Are we using the wrong approach? Maybe, or at least an incomplete approach. So it's remarkable to note that the development of GA therapy over the past 2 decades has been driven primarily by the tremendous genomic data that have influenced the field. And as Fede mentioned, in 2005, there were a series of landmark papers that identified gene variants in the complement factor H gene. So the complement pathway, as Dr. Rickman will no doubt tell you, is an important clotting factor mechanism, as well as an arm of innate immunity that has many functions. But complement factor H was statistically associated with macular degeneration, gene variants in that gene. And in subsequent years, many other groups identified in rapid succession many other complement genes that were statistically associated with AMD. In fact, high-risk CFH variants have one of the highest increased risks in a complex disease, in a multifactorial disease. Depending on which population is being studied, anywhere from 2 to 7-fold increased risk of AMD is found in patients with high-risk CFH alleles. But actually, if we look more carefully, what we find is if you look at all these different very large population sets, whether it's the AREDS data sets, the EU consortium, or these other data sets that you see, anywhere from 94 to 97% of patients who have the high-risk CFH alleles actually don't have AMD. And if you look further, it turns out that anywhere from 2/3 to 80+% of patients with AMD don't actually have this high-risk CFH alleles. And Rob Mullins has very nicely shown that children as young as 5 years of age have complement deposition in the back of their eyes and their choroid. And he's shown in patients who are in their 30s and 40s, long before they get AMD, tremendous amounts of complement deposition can be found. So I think it's fair to say that complement cannot be the sole explanation. It may have an important role. We don't know yet what it's doing in great detail. We certainly, the work of Dr. Rickman and many others have described us, the function of these genes and of these proteins in various contexts, but I don't think it can be the sole explanation. Yet it's also inarguable that the overwhelming research and development efforts in the field over the past 2 decades have focused on targeting complement. And these are some of the important trials that have taken place and reported results over the last 10 to 15 years. And these are all different companies that have used different strategies to block complement at various stages of the complement cascade, whether it's blocking C3 or C5 or complement factor B or restoring complement factor H levels. Until recently, there has been, unfortunately, a litany of failures. So it's really surprising and quite remarkable and intriguing that the Apellis group actually demonstrated the first phase 3 trial, which was the OAKS trial, that met the primary endpoint of reduced lesion size at 12 months. And subsequently, another company, Iveric, demonstrated in 2 different clinical trials that their complement inhibitor also met this primary endpoint. But it's also important to point out that these trials, the ones that have recently reported successes, have reported successes in terms of anatomy, not in terms of visual function. Whether it's pegcetacoplan, the C3 inhibitor from Apellis that is now awaiting FDA decision, or Zimura, the C5 inhibitor from Iveric that is supposed to be submitted for approval early next year. Both of these drugs have shown in phase 3 trials at various time points anywhere from 14 to 22% reductions in the growth of the GA lesion size. If you look at the final GA size difference, there's anywhere from a 4 to 8% difference between the treatment arms and the sham arms. Unfortunately, to date, there have been no visual benefits that have been reported in terms of visual acuity or microperimetry or reading speed, for example. And both these drugs have reported in various arms anywhere from 12 to 16% of patients developing what's called macular neovascularization or exudative AMD, which is about a 4 to 6-fold higher risk in those trials compared to the sham arms. So the summary, I think it's fair to say for complement trials is, to date, the majority of complement inhibitor trials have not met their primary endpoint, but very remarkably, some recent trials, like the ones I've mentioned from pegcetacoplan and from Zimura, have reported anatomic benefits, but to date, not yet visual benefits. And as I mentioned, we're all eagerly awaiting the decision, which may come as early as next month on one of these drugs submitted by Apellis. And the second drug is supposed to be submitted for approval early next year. So we may rapidly have one or perhaps two drugs that are now approved for geographic atrophy in the coming months. But I think it's overall fair to say the effects in those trials has been modest. So why might that be the case? I think it's because GA is like Murder on the Orient Express and not like Halloween, the movie. As you may know, in the movie Halloween, there's one crazed killer that's going around killing people. But in Murder on the Orient Express from Agatha Christie, there's actually a cast of culprits that are responsible for the death. And I think that might be what's happening in geographic atrophy. No doubt, complement has been implicated as an important risk factor for AMD. But several other groups, including ours, have shown that there's a multifaceted attack on the structure and function of the various cells in the eye that probably give rise to dry macular degeneration and geographic atrophy. Things such as mitochondrial dysfunction, retrotransposons, amyloid deposits, iron overload that Josh Deneve has spent his life describing, oxidative stress, and many other factors all are somehow involved at different times in different zones in different people's eyes. And I suspect that the cast of culprits culminates together in what we call geographic atrophy. Now, what's interesting is that over the last decade, numerous groups, including ours, including those outside the eye field, have studied these kinds of dysfunctions in many contexts, not only in the eye, but in the brain, as well as other diseases, and have remarkably described activation of something called the inflammasome, which is an arm of innate immunity that can respond not only to various invading pathogens in infectious conditions, but also to various danger signals such as these. And we think that inflammasome activation is an important aspect of geographic atrophy. So why did our group come to that conclusion? Well, we went back and tried to understand what is causing degeneration of the retinal pigmented epithelium, this important layer of cells that supports the cones and rods, the photoreceptors, and is interposed between them and the underlying blood vessel supply from the choroid. And these flat mount images of human eyes with geographic atrophy show the remarkable difference in the structure of the RP, which undergoes dysmorphology and degeneration compared to normal eyes without this condition. So we wanted to understand what's causing this RPE dysfunction. And to do that, we relied on a network of eye banks around the country that have been extraordinarily important in our work, as well as the work of many others in the field that have provided us this rich opportunity to study what's happening in the human condition. And then we've tried to understand what we find there in the context of various cell and animal models, and then go back to the human condition and see if the predictions from these preclinical studies are borne out in the human disease state. And recently, we've also coupled that with very large health data sets to try to identify clinical information that might be useful in trying to understand what drives geographic atrophy. And we've come to the conclusion over the past decade, as you see in the papers described there, that there's a multifaceted attack from complement and amyloid and iron and retrotransposons and mitochondrial stress and oxidized lipids that activate multiple inflammasomes, such as the ones you see described here, like NLRP3 and NLRC4, which result in the production of very toxic molecules called interleukin-1 and interleukin-18, which can cause cell death. And since our description of this back a decade ago, it's been gratifying to see the work of numerous others around the world who both replicated and expanded upon our own work in showing the importance of inflammasome activation in this disease. So we've been, of course, interested in how to block inflammasome activation. And several years ago, a very clever MD PhD student in my lab, Ben Fowler, and my colleague, Brad Gelfand, and others teamed up to find something remarkable, which is that a class of drugs that's already been used for decades in patients with HIV infection or hepatitis B infection called NRTIs, which are antiretroviral drugs, can actually block inflammasome activation, independent of their antiviral effects. Now, these drugs are pretty toxic. They're taken by these patients because they need to live, but it's really not appropriate to try to use these drugs for any extended period of time in patients who are elderly or who have many other disease states. So we created derivatives of these NRTIs called calmivudines, and one of these is now on the cusp of entering a clinical trial called SOM401. And what SOM401 can do is really remarkable. In various animal models, you can get RPE degeneration to occur using the various kinds of stimuli that you see here that we think are involved or can inform us about the development of geographic atrophy. And remarkably, SOM401 was able to block RPE degeneration, not just by one set of agents, such as agents that induce complement activation, but many others as well. Now, I mentioned to you that patients have been taking these NRTI drugs for 35 years. So we wanted to take advantage of the very large, big data sets that exist from various health insurance companies and other health organizations. So we actually worked together with a multi-institution consortium around the country to compile data sets of roughly 150 million patients and ask the question, are patients who are taking NRTIs at any reduced risk of developing dry AMD? And we published those data last year, and this is just one slide from that work, showing that patients who are taking NRTIs have a much reduced risk of developing dry AMD compared to patients who are not taking NRTIs. So while these data are certainly retrospective, they give us some information that corresponds to and is compatible with the concept that inflammasome activation might be important and that inflammasome inhibition could be valuable in preventing or treating dry AMD. And with that in mind,
Ambati, you're at the 18-minute mark.
Got it. Thank you. So as I mentioned, the company that I have co-founded called Inflammasome Therapeutics has now obtained approval to start a clinical trial of one of these inflammasome inhibitors, and we look forward to recruiting patients in the very near future this year. And various other groups have shown that inflammasome activation is actually very interesting from the context of many other aging diseases, not only macular degeneration, but Alzheimer's and multiple sclerosis and Parkinson's. And actually, there are 2 trials of inflammasome inhibitors that are ongoing now in Alzheimer's disease that should read out in the coming year. So we're all very excited about that as well. Of course, some of the work that I've described here has been made possible by our wonderful array of collaborators around the world, and nothing moves without money. And let me again reiterate my great appreciation to RPB for their support of my lab's work for nearly 2 decades, as well as the many other funding agencies that we have been very grateful to be supported by. But of course, most importantly, let me thank the amazing group of young scientists who I've been fortunate to be surrounded by over the past 2 decades, both in Kentucky and in Virginia. So with that, let me again thank Apellis for supporting this program and RPB for organizing this program. Thank you very much.
Thank you, Dr. Ambati. That was a wonderful talk, and give us that the importance of supporting basic research and research in general, to bring, what the targets we discover into, druggable targets into medicine. I think it was great. So now let me introduce the second speaker, Dr. Catherine Bose-Rickman, PhD. Dr. Bose-Rickman is the George and Geneva Bogus Ivansky Professor of Ophthalmology and a Professor of Cell Biology at Duke University in Durham, North Carolina. Dr. Bose-Rickman leads a team of researchers focused on the pathobiology of age-related macular degeneration and on the developing and testing therapeutic targets for AMD. Among her seminal discoveries was the identification of the gene responsible for retinal degeneration in the RD or RD1 mouse. In an effort to better understand the pathology of AMD, she has created a number of murine models that recapitulate many aspects of human AMD and point the way towards eventual treatments for AMD. These mice develop many aspects of the human AMD phenotype and provide an in vivo means to interrogate the pathogenic contributions of genetic, inflammatory, and environmental factors on AMD pathogenesis and test emergent therapies. Dr. Bose-Rickman has received 3 of Research to Prevent Blindness prestigious awards among her multiple awards and funding, and is currently a member of the National Institute Board of Scientific Counselors. Dr. Bose-Rickman's presentation today is complement-based strategies for age-related macular degeneration. Kathy, please go ahead.
Yeah, thank you for that lovely introduction. So again, thank you so much, Fede, for the introduction, and to Research to Prevent Blindness for the opportunity to give this presentation, and to Brian Hufflin. And of course, I am very appreciative of the fact that I've had 3 awards from RPB that have helped me through my career. They've been a great help, including the last one, which is the Catalyst Award for Innovative Research Approaches for AMD. Would like to quickly note my commercial disclosures and get started. So the outline of what I'd like to talk about today is to actually delve a little bit into the complement system, which apparently I'm doing for a second time. We'll do it a little bit more depth maybe. Come in and talk about the genetic risk and protection in AMD, drug trials for complement therapies, and insight from our clinical models. So what is the complement system? It's an arm of the innate, sorry, I have this in the way, an arm of our innate immunity that consists of a collection of proteins that circulate in our blood and tissues and that are ready to eliminate pathogens like a bacteria or virus. These complement proteins patrol our bodies in an inactive form, but are rapidly activated upon detection of a pathogen, culminating in killing it directly and/or helping immune cell recognize and kill the pathogen. So if we look at the complement pathway, there's actually 3 different ways to activate complement, but they all start with acting on complement C3. And this is a series of proteins that end up starting an enzymatic cascade that culminates on the cell surfaces where they're activated, bursting the cell. And so because this activation, this whole pathway can actually occur spontaneously, the C3 can cleave by tick over, there has to be a very strong system of regulation. And the soluble regulator of this pathway is known as complement factor H, which we heard about earlier. And this inhibitor will act to accelerate the decay of the C3BB convertase, which will block the rest of the pathway. And it also is a cofactor in the factor I-mediated cleavage of C3b into IC3b, again, stopping the pathway. So as we learned earlier, we know that back in 2005, there were 4 seminal papers that first described the risk association of complement factor H at the genetic level. But in fact, a number of years before that, there were many indications that this pathway and this protein were involved because of the fact that it was actually found in the deposits in the back of the eye known as drusen, and you can see that here. Since the discovery in 2005, we still don't entirely understand how factor H regulates the pathobiology, but we do know that it accounts for a very large fraction of the population attributable risk, particularly the variant that's known as a tyrosine 402 histidine variant, or the H402 variant. So as I mentioned, back there were many papers from Link Johnson and Don Anderson and Greg Hageman that were actually looking at what are found in the deposits. And they saw that by immunohistochemistry, one of the things that were commonly found there are components of the complement pathway. And here you see the inhibitor underneath the RPE, and then this hiding this sort of nidus of the lytic or activation factor. But as it's widely held that these genes are associated with risk, including the H402 variant, and pathology development. But there has, as we heard earlier, been some failures of clinical trials that were specifically targeting complement inhibitors that may diminish our support for this overall. In fact, it's possible that there's a non-canonical function of factor H that contributes to the drusen sub-RPE deposit biogenesis and the toxicity that contributes to AMD pathobiology. So one of the things that is important here is the effect, the overall effect of the risk. And it turns out that the two key loci associated with genetic risk are the first one on chromosome 1 that includes CFH and the downstream factor H-like factors, proteins, and then on chromosome 10, which is known as ARMS2 HTRI1. And these two alone account for almost 90% of the genetic susceptibility. While there are a number of minor or rare AMD-associated variants that are likely to modulate disease, these other loci have a marginal effect overall on assessing AMD susceptibility. And this is beautifully shown in a paper that was recently published from Greg Hageman's group, where you look at just the SNPs that define the genetic risk or protection around chromosome 1. And those are the risk allele, which is a Y402H, or the two protective alleles, which is an isoleucine to instead of valine, and then this deletion of FHR3 and 1 downstream. So these two are protective. And if you look at the effect of these together, you can see that the combinations are diplotypes that strongly influence AMD susceptibility, that the protective haplotypes by themselves can move susceptibility towards protection. So if you have two risk alleles or two H alleles, you have a high risk of getting the genetic risk of getting AMD. And then this is reduced somewhat if you just have one risk allele. But if you move into having any protective alleles, whether it's risk plus protection with I62V or the deletion, you now are overall, your susceptibility is now towards protection instead of risk. So what are the trials? I'm going to go into a little bit more depth than we saw in the previous talk. So if we look at the trials that have reached phase 3, and again, here's a schematic of how the pathway works. One of the first ones was against an inhibitor known against factor D, which is a important information of the convertase. And again, this is required for activating the pathway after C3. And this was the first phase 3 trial, and it failed in phase 3 to reduce GA growth. It was a factor D antibody that was delivered to the vitreous. But as we heard earlier, there is a pegocepticoplan or APL2 from Apellis, which is a pegylated peptide inhibitor that was based on an inhibitor from John Lambrish originally called Comstatin. And this in phase 2 showed nice 30% reduction in GA. But as it was mentioned, there was an increase in choroidal neovascularization in these patients. But now we have phase 3 and 24-month data that shows that both OAKS and Derby, which were the two clinical trials, met the primary endpoint of reducing GA, and that the CMV conversion was actually reduced compared to the phase 2 trials. Then, as mentioned, there was the C5 inhibitor, and this case it's an pegylated aptamer, again, a small molecule that's delivered to the vitreous, that also reduced GA growth by up to 30% in phase 2 and phase 3. In this case, there was also some conversion to CMV in a subset of the patients. And so, but at this point, at 12-month data reported that it met the primary endpoint of GA growth reduction. So diving a little bit more into the pegocepticoplan or the APL2 results, it did have clinically meaningful reductions in GA growth at 24 months. And what's interesting here is that the largest effect was seen in the last 6 months. And this has been a continuous trend, suggesting that over time, we may get even more reduction and potentially not just anatomical changes that result from this treatment. So there was meaningful preservation of the area around the fovea, and that the reduction in the meaningful reductions in GA lesion growth that demonstrated in both non-subfoveal subgroups, so these are patients who have geographic atrophy slightly away from the center, And then in patients who are right near the center, and again, with better treatment in the ones who got it every month versus, every month versus 2 months. But as was noted earlier, the visual endpoint changes did not change. And these are secondary endpoints, but there was no statistically significant difference across the study arms on key secondary endpoints at 24 months. And these include best corrected visual acuity, the maximum reading speed, functional reading, and microperimetry. And hopefully this will potentially change over time. It hasn't at this point. So some other studies, these are all studies, these next ones are actually antibodies that were used to target the complement pathway. The first was against C3, and this is the same molecule that is being targeted by the Apellis inhibitor, but in this case, in the form of an antibody, it failed in phase 2. It was delivered intravitreally, just like anti-VEGF is, which obviously works in wet AMD. And then there were 2 different antibodies that were tested against C5, so down lower in the pathway. The first was a systemic one from Alexion called eculizumab, and the other was from Novartis called tesidomide that was delivered intravitreally, failed. And then finally, there's an anti-preparidin, which is a molecule that stabilizes the convertase, acting higher up in the pathway to inhibit it, and it also failed following intravitreal with a monoclonal body, intravitreal delivery. So the last one here is actually an antisense oligonucleotide. Again, this is a small molecule, and it is to inhibit factor B part of the convertase. And it's still in phase 2, so we don't know. Some slightly different molecules. There are actually molecules, in this case, it is gene therapy, where they're trying to augment the amount of factor I, which is the inhibitor of IC3B, which acts in concert with CFH. And in phase 2, they found that it was well tolerated, and it actually increases the amounts of CFI in the back of the eye to 146%. There was another trial where they tried to augment the CFH protein itself by simply, they take the recombinant protein, this was Gemini, and they actually introduced the recombinant into the vitreous monthly. And unfortunately, this one had no effects at 6 months, and the trial has been stopped. Then finally, there's another regulator of the pathway. It's called CD59. It's a membrane-bound form, but they've made a soluble form that they're doing gene therapy with, and that's currently in phase 1. So what are the insights from our clinical models? When we look at our animals, we look for comparisons between the mouse presentation and the human presentation. So in humans, we see a reduced visual sensitivity, and we can see that by reduced changes in the visual function. We see RPE multinucleation and dysmorphogenesis in our animals, and then we also can see deposits in animals that develop the phenotype. So we tested the hypothesis that if we took mice and they were made to express the human AMD risk variant of CFH, that they would develop an AMD-like phenotype, but compared to the normal variant would not. And this would give us in vivo proof that that variant by itself can contribute to AMD pathology. So we generated transgenic mice, and these were mice that expressed the full-length CFH gene and nothing else on either side. And they either had the normal variant or the risk variant, and they were crossed onto a knockout background. So they only make the human form. When we originally generated these animals, the H form only made about half as much as the normal Y form. So we simply bred them together, and they both make the same amount of these human complement proteins. And interestingly, or just as we'd hoped, they do actually work in the mouse pathway. And this is illustrated here. The knockout mice have no factor B and no full-length C3 because it's functionally cleaved by the loss of CFH. It's all consumed, but you can see restoration of factor B in both the Y animals and the H animals. And it's noteworthy that they both have the same amounts. There's no difference in their complement system. So what we did is to test this in all of our models. We aged the mice so we can replicate aging. We switched them to a high-fat cholesterol diet for 8 weeks, like living on a McDonald's diet for 8 weeks. And then we look at the changes in visual function and other things. And what we saw in our animals is that we saw a change in the visual, a decrease or attenuation in vision. And this only happened in the H animals with the risk factor with the HFC diet. So now we have a model where we can actually look if we can rescue this model with therapies. But when we look further into the animal, we also saw that there are changes to the RPE, just like we see in humans, where there's more damage in the H mice that have been fed the diet. As I noted before, there's no change in the complement pathway in these animals. What you see here with all the R's is the green ones are all higher. And these are both Y and H animals. And it's an overall systemic response to the high-fat diet because it stresses your body. So you can see in the plasma that you have increased complement factors, but also in the eye, but they're all changed to the same amount. There's no difference between the Y and the H animals. However, if you look in the eyes of these animals, there is an increase in lipoproteins that's specific to the H risk animal. And that's an increase in ApoA1, which is very interesting because this is a known lipoprotein that is associated with AMD risk. And in fact, if you look at, instead of being the good lipoprotein in like it is for cardiovascular disease, it's actually associated with higher risk in AMD. And when we looked at this a little closer, we looked at human donors and we looked at the back of the eye at the HDL present there and compared that to the plasma in normal, in the same donor. And what's very interesting is it turns out that in the lipoproteins that are in the back of the eye, there's an increase in the amount of HDL. But what's really striking about that is that this is HDL that is much more pro-oxidant and pro-inflammatory. So what's sticking in the back of the eye is actually a nidus potentially for more inflammation. And it turns out that CFH interacts with this molecule. In fact, if you look at this is again another human donor eye where you have the different kinds of lipoproteins in the back. And if you add the Y form or the normal form, it does a great job of taking all of the cholesterol, different moieties, lipoproteins off of Brooks membrane, which is where it resides in the back of the eye. But the Y form, the risk form does not do that as well, especially related to the HDL. So just as we saw in the mice. So the thought is that there's this intersection between the lipoprotein pathways and complement regulation that may actually be important. So what we're in the process of doing now is augmenting complement factor H as an AMD therapy. So again, but doing this from a gene therapy perspective. So the arguments are that we know that as I showed you earlier, that the haplotypes, the protective haplotypes, and the SNPs actually will mitigate the risk of the risk form, in not only on chromosome 1, but also with the ARMS2 HDR1 locus, which was another big surprise. That the factor itself, the risk variant, is in the sub-RPE choroidal space as an initiator of dry AMD pathology, which I just showed you with our animal model. And that we believe that augmenting CFH should correct complement dysregulation in addition to any non-canonical interaction of CFH with the protein pathways. And finally, that in contrast to simply or just inhibiting C3 or C5 overall, that the CFH augmentation should not perturb any homeostatic role for ocular complement. And that hopefully that this would now protect it instead of just inhibiting it outright, since we know that's needed there. So what are the hurdles for augmenting CFH as an AMD therapy? Well, we first had to figure out whether or not you want systemic or local delivery. And absolutely from our work, we know that it needs to be local and to target the RPE Brooks membrane. So to get to the basal side of the RPE, not delivery from the vitreous. And that it turns out that CFH is quite a large glycosylated protein, so it does not readily cross Brooks membrane. This is what was elegantly showed by the work of Simon Clark. And that therefore, we're trying to design forms of the CFH that are slightly smaller, but still do all the same things. So this would be like the mini factor H that was designed originally by Lambris and tested in vivo by Marchbank and colleagues. And then the stage. So right now we're all focusing on GA. It's sort of FDA regulated as well, but we actually think both at GA and intermediate are the best stages to study for or to treat. And then the formulation. And we're working on a gene therapy. This also based on the fact that a protein delivery, when we saw in the earlier studies, delivered to the back of the CFH by itself was not able to rescue. And I think that has a lot to do with the fact that it probably didn't have access to the basal side of the RPE where it needs to function. So with that, I would like to finish and of course take time to thank all the people that actually do this work. I get the opportunity to talk about it. I have a wonderful group in my lab, both present and former members, great collaborators at Duke and outside of Duke. And I would also, of course, like to thank my current funding support. Thank you very much.
Thank you, Dr. Rickman. That very stimulating talk. A lot to think about it, and looking forward to what this will bring. Now we do have I had some questions, but I'm going to go to the question that we have in the list. I'm going to rephrase it a little bit. So the question was whether there is any treatments or cure in the future for AMD and geographic atrophy. So I'm going to answer the first part. There are several treatments for wet AMD, all within the anti-VEGF space or mode of action for the time being. There are no approved treatments for GA, but as Dr. Ambati said, we are excited about the possibility of having the first treatment available soon. But I mentioned at the beginning, and you both reiterated the multifactorial nature of this disease. So with that in mind, do you see any cures, and by cure meaning perhaps stopping or reversing this disease in the horizon? And I'll throw it to Jay first, and then you, Kathy.
Sure, we can cure this. It's very simple. All you have to do is delay the development by 13 years. Okay. So, if you to put it very bluntly, if the patient dies before they get geographic atrophy, you've cured this disease, right? The median age of GA development is roughly 73 years of age. The median lifespan is roughly 78 to 80, depends we're talking before or after COVID, right? So if you delay by a dozen years the development of this disease, you can cure it. So that's not just a tongue-in-cheek response. There's lots of people around the country and around the world who are working on basically treatments for aging, right? There are entire companies based on that. And there are many approaches. One of them, of course, we're tremendously interested in is something called inflammaging. Vishwa Dixit has done a lot of work on this concept of inflammasome in aging. And there are also companies working on retrotransposons and aging, and there are any number of approaches. So yeah, I think it's quite conceivable that if you have treatments that delay functional aging, we can cure GA functionally.
Kathy?
To add to Jay's response, I think that it will be nice for us that the idea of trying to develop a gene therapy based approach is sort of a one and done. So if we ever get to the point where we can actually do that, then we will be able to potentially arrest or stop the changes with in this case, the CFH augmentation. And then, it's not a repeated therapy. So that's our biggest hope. And of course, we've already got from your company good evidence that the complement pathway is a great target. So we just have to see how to tweak it or what kind of combo we need to do to make it work.
All right, thank you very much. So now this one for you. So first of all, I found the CFH hypothesis very interesting and very encouraging for the future. If we could increase really protection, that'd be wonderful. Now, if we go back to the presence, and of course, a cure may be coming, right? But what we have right now in GA are some of the complement inhibitors that have worked, and some others have failed. Do you have any hypothesis on why that is the case of what we have seen so far, right? From the studies on why some parts of the pathway may have, not work. So Kathy first, and then Jay, if you want to add anything.
So I think, and I tried to break up the description of the therapies in that way. I think the thing that's really striking is that there's been basically a total lack of progress or failure for every single therapy that was based on antibodies. whether they were inhibiting the pathway systemically or intravitreal. And I think, one of the things we sort of lose sight of is that it's absolutely true. The anti-VEGFs are a miracle drug for wet AMD, but this is a lesion that's basically creates a hole in the back of the eye. And I think the access to the subretinal, to the sub-RPE for antibodies in the vitreous that in a GA eye the dynamics are totally different. And this is borne out by the fact that none of these trials have shown, and it's interesting because you have the head-to-head of the anti-C3 that was intravitreal against the Apellis in C3 inhibitor, where one did a marvelous job of reducing GA progression, and the antibody version of the anti-C3 did not. And again, that's one that's being delivered from the vitreous, and then the problem, of course, is systemic. Even which it didn't work, you wouldn't want to give a systemic treatment for an antibody treatment for this disease, and I'm sure Jay doesn't disagree with that. So.
Jay?
Yeah, so I'm a very data-driven person, and so to me it's puzzling that some C3 inhibitors have shown to meet certain endpoints in terms of anatomy, and others have not. Some C5 inhibitors have been shown to have benefit in a clinical trial setting, and others have not. It's speculative to attribute the failure to, let's say, antibodies or mode of delivery. If you look at, I think work from a lot of groups, whether it's from Hageman or Mullins or lots of other groups, have shown that the bulk of complement, especially MAC deposition, is in the choriocapillaris in patients with this condition. So one might have thought that the best complement inhibitor known to humans, which is eculizumab, and your drug, which I'm actually blanking on. It's the same name for that also? I thought it had a different name. Okay.
No, it's the same, the drug
for
name.
PNH. For paroxysmal nocturnal. Okay, so it's the same name, so pegcetacoplan. And so if you look at Solaris, it blocks 99.99% of complement activation in humans when delivered systemically. Phil Rosenfeld showed several years ago that at least at 6 months, there was no effect. Now, will it have an effect at 6 years or 60 years? You have to do the trial to find out, right? But the data that we have is that the best complement inhibitor delivered systemically, which no doubt gets to the choriocapillaris, wasn't able to show a benefit. Whereas in one of your trials, you were able to show a benefit at the primary endpoint. And in the context of C5, they did. So, and more to the point, ultimately, whether it's the FDA or patients, they don't really care about mechanism of action. They care about action, right? And so if the risk-reward ratio is beneficial, it doesn't matter, how the drug is working and how it's delivered. And, time will tell us more with more of these complement inhibitors, and hopefully the field will test drugs apart from complement inhibitors as well.
Yeah, I think that's very important and very likely, and that's where we need to go again. Going back to the multifactorial nature, I don't think there's a single target. I think we need to go, broad to your point. So now, going back to, delivery, and we say, of course, for certain drugs we don't want to go systemic, but as we look at the future and those cures, do we still think local or we think systemic? We have 2 minutes left, so I don't know who wants to go first, but quickly, you have 1 minute each.
I think, obviously for systemic, the risk profile has to be tremendously different, right? So the attraction for local delivery is precisely that, local delivery with reduced systemic exposure. But if you have a drug that's safe systemically, I think in many ways it might be beneficial for patients to simply pop pills at home.
I would have to agree with that. It would be great if we get to that point, maybe even with your drug. But I would remind you that in the case of whether it's in the choriocapillaris or in Bruch's membrane on the RPE side, there's certainly complement activation there and local complement synthesis. That's very important. And from the perspective of the fact that the systemic form didn't work, further supports that it really needs to be, in the Bruch's mem, you and I both know, all three of us, that it's a pentonominal region and that there is this division, and what's closer to the RPE could be what we need to be targeting, and that's not happen. Wouldn't happen. It's certainly not, with these big molecules systemically.
All right. Thank you both so much. Thank you all the attendees and RPB for the opportunity. And Diana, back to you.
Yes, thank you so much to all of our speakers, Dr. Grossi for your excellent moderation, Dr. Embody and Dr. Bose-Rickman for your fantastic talks. We want to thank Apellis again for generously making this event possible. And finally, thank you to everyone who is watching and who has joined us for this Lunch and Learn. We hope you will join us for future events. And we look forward to sharing more information with you on the exciting research that is occurring out in the field and that we hope will soon be at an ophthalmologist's office near you. So thank you so much, everyone. Have a great day. Take care.